Hello, everybody. Welcome to the Stetten lecture. As we begin, please note this lecture is being recorded. My name is John Lorsch. I'm the director of the National Institute of General Medical Sciences. Welcome to the DeWitt Stetten Jr. lecture, which is part of the NIH Director's Wednesday afternoon lecture series. The series honors DeWitt Stetten Jr., who was the third director of NIGMS and had a strong commitment to basic science. He spent most of his career at NIH serving in 2 institutes as well as the Office of the NIH Director. And he was particularly well known as the co-author of the early editions of the textbook Principles of Biochemistry. In recognition of his many contributions to NIH, both within and outside of the NIH, he has the rare distinction of having 2 NIH entities named after him, this lecture and the DeWitt Stetten Jr. Museum of Medical Research, which you probably actually saw when you walked in because it's distributed around the campus, most of it, I think, actually in this building. So the exhibit out there, as well as the one around the corner of Marshall Nuremberg and the discovery of the genetic code is part of that museum. The way the lecture works is that the speaker, Dr. Sudha Chakrapani, will give a 50-minute talk on her research, followed by a 10-minute question and answer session. You can ask your questions by using the mics in the room, if you're in the room, or by selecting the Send Live Feedback button on your videocast screen if you're online. If you are seeking a continuing medical education credit, or CME, the code is 57663. I'll repeat that, 57663. We invite those of you here in person to join us outside of the amphitheater for a brief reception after the lecture, just right out there. So, today's speaker, Sudha Chakrapani, joins a very distinguished list of Stetten lecturers. She is a professor and chair in the Department of Pharmacology at Case Western Reserve University, and she serves as the director of the Cleveland Center for Membrane and Structural Biology. Sudha has dedicated her career to investigating ion channels that mediate fast synaptic transmission at the neuronal and neuromuscular junction. She leads a laboratory that utilizes cryo-electron microscopy to investigate ion transport in healthy and disease conditions and how medicines can target it to treat specific diseases. During today's lecture, Sudha will discuss glycine receptors, including the conformational changes they go through based on different factors, their regulation, and the potential to develop subtype-specific therapies for various disease states. Sudha received her master's degree in biomedical engineering from the Indian Institute of Technology and a Ph.D. in biophysics from the University of Buffalo in New York. I'm pleased to note that NIGMS supports Sudha's work through our Maximizing Investigators Research Award, or MIRA program. And now, please help me welcome Sudha for her talk, Molecular Mechanisms Underlying Glycinergic Neurotransmission. Sudha.
Bryce, I'm good? All right. Good afternoon, everyone, and thank you, John, for the very kind introduction and for the invitation to present the Stetten lecture here today. It's a tremendous honor for me to be here and to also present some work that we've been doing in the last 10 years or so that has been continuously funded by NIGMS through R01 and R35 awards. I must also mention that our work, which heavily relies on cryo-EM as a technique, has been supported by the infrastructure at the national centers that funded by common fund programs and now through NIGMS. So delighted to share this work with you, and thank you all for joining me here today. So what I want to tell you about is somewhat of a journey that I've had through studying how chemical synapses behave and what are some of the intricate underpinnings of receptors that mediate fast communication. So these receptors, the ones we study, belong to a family of pentameric ligand-gated ion channels. They govern a wide range of physiologies, from muscle movement to learning, memory, gut-brain signaling, pain perception, and so on. Amongst this class of channels is glycine receptors, which I will be talking about today. And since I started, my master's degree and going through my graduate education, this journey of studying these receptors has taken me through a wide range of approaches. Started as a single-channel electrophysiologist, then moving on to structural biology, studying X-ray crystallography, EPR, electron paramagnetic resonance spectroscopy, and now cryo-EM. So it's been a fun ride so far. So on to glycine receptors. So these receptors play a very important role in regulating sensory pathways and motor coordination. The signal that comes from, the signal that comes from the peripheral terminals brought in by nociceptor neuron or sensory neurons eventually go to the somatosensory cortex where it is perceived as pain. And this signal is regulated in the dorsal horn of the spinal cord by the glycinergic receptors right here. Similarly, the signal that comes from the brain that goes to the motor neuron that mediates muscle contraction again is regulated by glycine receptors. So let's zoom into this region of the spinal cord right here. So the signal brought in, so the signal that is brought in by the peripheral terminals eventually goes to the dorsal root projection neuron carried through the spinothalamic tract to the brain. And this signal is modulated by inhibitory interneurons that release glycine and GABA as their neurotransmitters. These neurotransmitters diffuse across the synaptic cleft and bind to the glycine receptors on the postsynaptic membrane right here. So as you will note that these receptors actually come in two flavors. They are either on the postsynaptic membrane, they're also present in presynaptic and extrasynaptic spaces. So the glycine receptors have a slightly different architecture that populate these spaces. They mediate tonic inhibition. And whereas the receptors on the postsynaptic membrane, they appear to be more clustered by a very exquisite mechanism that I'll touch upon a little bit later. And these maintain the postsynaptic inhibition. So the glycine receptors, there are 4 genes that code for the alpha subunit and 1 gene that codes for the beta subunit. So the alpha subunits are the ones that form functional channels, the alpha 1, 2, and 3 subtypes. These subunits can assemble on their own to form a homopentameric channel. So the homopentameric channels are found in the presynaptic and extrasynaptic membranes, whereas the beta subunit, it cannot form a pentamer on its own, so it assembles with one of the alpha subunits, and the beta subunit is part of the assemblies of glycine receptors in the postsynaptic membrane. And the reason for that is that the beta subunit has a sequence that binds to a protein called gefrin, which is a postsynaptic scaffolding protein. So this protein is essential for the postsynaptic architecture. Looking at it at the individual level, each of the alpha subunits share a very strong sequence similarity, and including the beta subunit, this is about 85 to 90% sequence similarity, and in the beta subunit, you have about 50% sequence similarity to these. So each of these subunit, as you can see, has 3 modular domains. The extracellular domain, which houses the neurotransmitter binding pocket, it is set of beta strands that fold to form this scaffold. The transmembrane region has 4 helices, and this region is the site for selectivity. Glycine receptors are chloride selective channels, so it's a site for selectivity and its gating mechanism. And a region that we don't see here in the scaffold is the intracellular domain. And we think that this is a very interesting region, but structurally really hard to work with, because this is highly unstructured. And what you will see in the slides that I'm going to show are we haven't really been able to get a handle on this domain. But what makes them very interesting is that this intracellular domain is the region that binds to gefrin that I mentioned, the postsynaptic scaffolding protein, but it also interacts with several other proteins that post-translationally modify these receptors and modulate their function. So there is a lot that we need to still do to understand this domain. Also in this, I wanted to mention that the alpha subunit, alpha 1 type, is expressed throughout the gray matter of the spinal cord, whereas the alpha 3 subtype is only present in the dorsal horn. And again, the alpha 1 brings about the motor functions, whereas the alpha 3 is involved in pain perception. So they have distinct expression patterns and functional behavior. So the alpha 2 is the first of the glycine alpha types to be expressed. They are expressed before the synapses are formed, and they play a very important role in the interneuron migration. And these, if alpha 2 is knocked out, the number of dorsal projection neuron numbers is pretty low. And so they play an important role in having synapses develop. And another interesting feature is that during development, the chloride gradient is actually opposite. So when these channels open, they depolarize the membrane. So they depolarize the membrane, activate calcium channel, and eventually causes the actin-myosin engagement and leading to neuronal migration. But as the synapses develop, you have this exquisite architecture that I was talking about, which is an interaction network. You have glycine receptors interacting with gephrin, which in turn interacts with colibestin, and then actin and tubulin. And so this is a network of proteins that maintain the synaptic architecture. And also, what we see here is that the clustering of these receptors in the postsynaptic membrane is activity dependent. So if you block the functionality of glycine receptors, you see that these receptors diffuse away and the synaptic architecture is modified. So there are a number of mutations that are known in glycine receptors that impair glycinergic neurotransmission, and many of these are in the alpha 1 subunit. Its dysfunction is associated with a disease called hyperekplexia. It's a neurological rare disorder. In this situation, the babies experience an exaggerated startle reflex and seizure-like symptoms. And if you look at where these mutations are, they are located throughout the protein. And most of the impairment that you see comes from a decreased glycine receptor expression, an increase in desensitization rate. You see a reduction in glycine sensitivity and chloride conductance, and also a loss of zinc potentiation, which I will address in this talk today. Another kind of neurological disorder is a stiff person syndrome disorder, and another one which is the progressive encephalomyelitis, which is a severe form of the stiff person syndrome disorder. In this case, the person makes antibodies towards glycine receptors, and as a result, you have a drop in glycine receptor density in the postsynaptic membrane. Pathophysiologies that are associated with alpha 3 glycine receptors come from their role in inflammatory pain states. So during inflammation, cyclooxygenase, which produce a prostaglandin, which activate prostaglandin synthase, and eventually release of prostaglandin activates the prostaglandin receptors, which activate cyclic AMP production, and cyclic AMP then activates for protein kinase A, which phosphorylates alpha 3 glycine receptors. Phosphorylation of these receptors then decreases or diminishes glycine receptor function. As a result, you have hyperactivation of these neurons and transition to pain states. To support this model, a phosphodeficient mutant that does not carry this serine, which is phosphorylated by pKa, you can see that the impact by these mice lose a sensitivity to prostaglandins. So in terms of pharmacology, glycine receptors have a very diverse and rich pharmacology to take a look right here. So there are a number of known binding pockets which are modulated by a range of endogenous and exogenous ligands, and many of these may have are of therapeutic value. So there are ligands that were pursued by Amgen and Pfizer. These are sulfonamide-based compounds that bind to the extracellular domain. There are cannabinoids, both endo and phytocannabinoids, neurosteroids, alcohols, ivermectin and anesthetics bind to these various pockets, and also modulated by endogenous regulators like pH and zinc. The issues that we face with using any of these right now is that these compounds are fairly non-selective for glycine receptors. They target a wide range of receptors. So this is a good starting point. It's a great starting point to understand the fundamental mechanism of how these modulators affect function, but we really are on the lookout for better modulators of these receptors. So what do we know about the different subtypes that we can harness to target them? So the alpha receptor, even though they have pretty strong sequence similarities, you can see that they have differences in sensitivity to glycine. As you can see, alpha-1 is much higher sensitivity to glycine and also to partial agonists compared to that of the alpha-3 subunits. You can see that from the shift in the dose response. We also see that the assemblies, the homomeric receptors and the heteromeric receptors also have differences in sensitivity to glycine, as you can see in the shift in the dose response. Not only that, they also display very distinct single channel behavior. You can see the alpha-1 populates at least 5 different subconductance states, whereas the alpha-1 beta heteromeric receptor has a shift towards lower subconductance states. So the questions that the lab has been exploring in the last few years and areas where we want to head towards. First off, we want to understand the structure and dynamics that underlies fundamental mechanism in these receptor families. So we study glycine receptors, we study serotonin receptors, and other member of the same family. We want to really understand what goes on at the molecular level. We want to understand, when we study these by cryo-EM at this time, we are still looking at a minimal unit. We are looking at a purified receptors. But these receptors seldom work alone. They are a part of supramolecular complexes that modulate function. So we want to be able to eventually achieve understanding how these receptors are assembled in their functional entities. And finally, we want to be able to look at them in their native cellular environment, in this case, in the context of a synapse. And I mean, this is a stretch goal at this time, but hopefully we as a field will get there sometime. So what do we know about the structures of these receptors? So the pioneering work in the glycine structure field was done in the labs of Eric Guo. This was a cryo-EM structure going back to 2015 of alpha-1 glycine receptors. And at the same time, the group of Paul Schaefer from Amgen, they published this X-ray crystallographic structure of the alpha-3 glycine receptors. So early efforts to capture the structures of these receptors involved truncating the intracellular domain. As you can imagine, problematic domain, wouldn't crystallize the protein with that. So these were the initial efforts, and these within these structures, they were able to capture multiple conformations. There were some lingering questions that were eventually answered with structures published by our group. So these and Eric at Guo's lab at the same time. So these are full-length receptor structures. This is reconstituted in lipid nanodisk, and in this one in SMA polymers. So again, these are both full-length structures, but as you can see, we haven't still gotten initial view of the intracellular domain. And more recently, last couple of years, there are a number of heteromeric glycine receptor structures from Wei Wang's group at UT Southwestern, and this is a structure of a heteromeric receptors from Eric Guo's lab, from native sources, from pig brain, and this is from our lab, heterologously expressed. At this point, the stoichiometry, how many Bs and how many As constitute these receptors, was still unclear. 3 labs, 3 independent approaches, 3 expression systems, arriving at the same conclusion of a 4:1 stoichiometry, 4 alpha and 1 beta, and I think that was quite remarkable. Another point to note here are in this structure, they used GFP, so they removed the intracellular domain from beta subunit and replaced it with a GFP. So that allowed them to annotate which one is an alpha and beta subunit. In this case, Eric Guo's lab used an antibody which recognizes the alpha subunit, again used as a fiducial marker for annotating subunits. And in our case, we actually used gefrin as a co-complex, and we were able to image this without additional fiducial marker, and the protein had enough signatures within it that allowed us to solve the structure independently. So our lab studies each of these subtypes. There are interesting questions that are unique to each of these subtypes. But what I want to do today is just show you 3 vignettes that captures how are these receptors activated by glycine. What are the, which I chose to talk about 2 endogenous modulators, zinc and pH, in the context of pain. I want to show you cannabinoid modulation in the alpha-1 subtype. And I want to finish off with modulation of alpha-1 beta by ivermectin, which is a potentiator of this channel, to show you a glimpse of how we can sort of tackle subtype specificity. So let's see. At a fundamental level, these receptors will cycle through these gating schemes. So there's a resting state, getting activated by an agonist. The channel is transiently open. You can see this current goes and then it desensitizes. So it gets into the desensitized state. But this is a pretty big oversimplification because these, as you could see it in the single channel conductance, there are a number of states that these channels populate. But at least this is a starting model for us to go. And what do modulators do? The allosteric modulators essentially switch the equilibrium between these states. So if you're using a potentiator, it pushes the equilibrium towards the open conformation. If you're using an inhibitor, it perhaps shifts it towards a resting state. And this is sort of a theme that you will see in the data that I'm going to present. So this is work that was done by a graduate student, a very talented graduate student in the lab, Kayla, and a really talented postdoc, Eric Gibbs, in collaboration with Phil Biggins' group, David Seifert, who was a graduate student in Phil's lab. So all of the molecular dynamic simulations were done in Phil's group. So we wanted to study zinc modulation in alpha-3 glycine receptors. So zinc is co-released with synaptic vesicles containing glutamate. They have a number of modulatory effects on synaptic receptors. They go through all of the mechanisms, through phasic modulation, through tonic modulation, and also through this diffusion of zinc across to other synapses. So glycine receptor has been long known to be modulated by zinc, but what is interesting is that unlike other receptors where they inhibit, but the effect of glycine on alpha-3 is biphasic. So at very low concentration, and are close to 1 to 10 micromolar, up to 1 to 10 micromolar, you have this potentiation. Whereas beyond that, you start to see this inhibition. So right off, we know that there are 2 binding sites, one that is potentiating, high affinity, the other one is low affinity inhibitory sites. So mutagenesis have given us a glimpse of where these sites might be, but just that there are many sites outside of these that also impact zinc modulation. And so what are allosteric and what really are zinc binding sites were still questions to be addressed. The other thing from a disease physiology is that mutations that eliminate zinc potentiation have hyperexplexia phenotypes, suggesting that zinc modulation might be a physiological mechanism of how these receptors function. So to do this, what we expressed alpha-3, full-length human alpha-3 glycine receptors. We purified them using affinity chromatography. We reconstitute them in peptidisc. It's a belt of peptides that wrap around the transmembrane regions. So then we image these, and then you can see these images that are aligned, showing side views, top views of the protein. You can start to see some helices. This is where you get really excited because you start to see helices at this 2D positions. Then you can take these particles and classify them, and you will see in this data set that within the same data set, we start to see multiple conformations that are there that the algorithms can sort it. So our strategy was to image this under lower than the EC50 for glycine, so that you are capturing some non-activated and activated population. So what we saw, which is what we are seeing here, so this is the current traces for glycine, and here we imaged it under 100 micromolar glycine concentrations. And once you sort the particles, we see that the particles belong to 2 different conformations. I'll tell you what those are roughly at 50% occupancy into these 2 states. So this is the map of those 2 conformations. This is the model that we built, and what I'm showing you here are 2 subunits. This is sliced through the channel to see the ion permeation pathway. So this is a resting conformation. You can see that the channel is So the green patch right there is where an occlusion occurs of ion permeation. I can see that it's a constricted radii plotted along the length of the channel. And this is a ring of leucine residues that constrict ion permeation. So this forms the activation gate of the channel. In the in the desensitized conformation, what we see is another, so this gate right here is wide open, whereas this gate right here is constricted. It's a ring of proline residues that causes constriction in the activated, once the channel is activated. So zooming into the binding pocket right here at the interface between these two subunits, we can see that in the resting conformation or the closed state, you see no density. It's an empty pocket. Whereas in the glycine-bound state, you have a nice density, and you can map out all the interacting partners that interact with glycine. So what happens is glycine binds here, and this loop, which is referred to as loop C, closes in onto the binding pocket and causes a twisting movement that is eventually conveyed to the pore of the channel. So that's the activation mechanism, a mechanism that is very conserved amongst this family of channels shown by our lab and several other labs. Then what we did is then we increased the concentration of glycine. So we went up to saturating levels. And what we see is that the equilibrium shifts towards the activated state right here. And the state is very similar to that of the state captured here. So here is a movie that summarizes the movements that we see upon glycine activation. So what you're seeing here is the glycine pocket. You see this inward movement that I talked about of loop C, and that's a little glycine density at the back end. And once glycine binds, so glycine binds in this pocket right here. So these are the interacting residues. And that's the inward movement over the glycine. And this is an iris-like movement of the channel pore. So this allows for ions now to pass through. How is this basic mechanism of gating or glycine activation affected by zinc at low concentration? So here is the zinc potentiation that you can see at low concentration. It sort of tapers out and starts to move into the inhibition mode right here at higher concentration. So then we image this in the same condition, what we are looking at. So this is in the absence of glycine. Again, you have these distribution. And then when we add 1 micromolar glycine, which is when you're potentiating, we are simply eliminating this closed conformation. You really have all of the particles appear to belong to the desensitized state right here. And then if we go towards the higher concentration of glycine, we shift the equilibrium towards the closed conformation. So you can see that these states are very similar to glycine-bound states that we saw before. The zinc's role has been in shifting these equilibrium. So we ask the question, what is driving this shift in equilibrium between these populations? So to summarize, actually, so here we have we have identified 2 zinc binding sites. The potentiating or the high affinity zinc sites that are occupied at really low concentrations of glycine is right at the interface between the extracellular and the transmembrane domain. And you will see in the next slides that this position is optimally placed to interact or regulate the movement of loop C. So this allows for loop C movement to happen that facilitates glycine-mediated activation. And the second binding site for glycine, which is the low affinity inhibitory site, is at the back end of the pocket. So this is the high affinity binding site. We can clearly see it at this interface. There are 4 amino acids that coordinate the zinc, 2 aspartate glutamate, and then the 2 histidines. So they belong to different beta strands here and the M4 helix right at this. So at an optimal position to couple movements in the extracellular and transmembrane domains. And this is the inhibitory site that we see at the back end of this pocket. Unlike the high affinity site, which has a tetra coordination, the low affinity site is coordinated by a histidine and a glutamate, and the remaining 2 likely through water molecules. So so this is a site that is accessible. So its accessibility is does not underlie low affinity. It's just that the coordination is weaker than the high affinity sites. So here is a movie that highlights the zinc sites in both the closed and the desensitized conformations. So that is a zinc coordination for the high affinity site. So this, the traces are in orange are the resting conformation, and the blue is the activated state. So there is a slight rearrangement between the two states that seem to suggest that zinc in the activated state is better coordinated, and this is eventually translated to loop C moving inward, and this is the back end. So this was a tricky study to understand why this would lead to an inhibition at this pocket. It doesn't seem to be very close to it's behind the binding pocket and cannot directly influence the binding site. But digging deeper into the literature, it was clear that this arginine and glutamate, they make a salt-bridge interaction, which is very crucial for glycine activation, and mutation to these residues have been associated with hyperexplexia. So this interaction needs to be in place for glycine-mediated activation, and we think that having a zinc in this coordination triad is weakening the interaction between arginine and glutamate, and that might mediate inhibition. So we collaborated with Phil Biggins' group. They evaluated the stability of these interactions by long molecular dynamic simulations, and they find that zinc throughout the simulation in the potentiating site stays pretty close. These residues interchange in terms of their interactions, but the zinc itself stays firmly bound here. They also saw that the glutamate was an important interacting residue for the inhibitory site, and again, stable interactions were noted for the glutamate and zinc right here. So again, comes to our proposed mechanism that you have this interface interaction that mediates loop C movement, and which might be stabilizing glycine in the pocket and increasing affinity, and inhibitory site might be impacting glutamate-arginine interaction. To further look into whether how much this proposal of this idea holds, so Phil Biggins' group looked at the interaction during the simulation in coordinates where there is no zinc versus those with zinc. So this interaction is very stable when there is no zinc, and then when you add zinc, these residues start to move towards zinc and weaken the interaction, and so we think this could be a starting mechanism. We also, structures, these are static images, so these mechanisms need to be validated. In this case, so this is looking at zinc modulation. So this is in blue is the potentiation window, in orange is the inhibition. We tested the hypothesis by making mutation, essentially deleting this end of M4, where there are multiple residues that were participating in this coordination, and we see that zinc potentiation completely goes away. Similarly, we make a mutation to this glutamate, and we see that the inhibition gets shifted to much higher concentrations. So this sort of gave us some idea on what's happening with zinc modulation. So another modulatory factor that is important in pain sensation is pH. During neuropathic pain and hypoxia, so you have the extracellular pH moves towards acidic direction, even to some extent to 1 pH unit. And previous studies have shown that under acidic pH, glycine sensitivity is right shifted. Also, its activity to partial agonism is decreased. It is very interesting to note that at acidic pH, what we are essentially seeing is a reduced glycinergic response based on the peak amplitude, faster decay. And at the single channel level, under acidic conditions, you have these very short-lived closures. So the channel tend to move into the closed conformation, multiple types of closed conformation. So you have one that is coming from reduced amplitude, but also these fast flickers that are contributing to lower open probability. So we then looked at how alpha-3 behaves under these acidic conditions. So here is the electrophysiology showing inhibition. But like other modulators, at saturating conditions, you diminish the effect of this modulator. So what we noticed is under acidic pH, so this is under normal regular conditions at 100 micromolar glycine, pH, lower pH populated a number of states that had distinct features that were different from that of these conformations. So the shift was, the first thing we noticed, a shift towards more closed and non-conducting conformations, and also appearance of a new intermediate state. And again, when we move towards saturating concentrations, all of these states shift towards the desensitized state. So here is a movie that shows you what these changes are. So the closed conformation that we stabilize has a different orientation for loop C. It also, interestingly, has glycine in the pocket. So you have glycine, yet the channel is not activated. And you can see some rotameric differences. This is the intermediate state. Again, you have glycine in the pocket, the channel you will see. So this is moving towards glycine activated state. And here is a desensitized conformation. Here is the intermediate conformation, and then moving to the closed conformation. So we have the pore of the channel is stabilized in three distinct states that contributes to non-conducting states, and we hypothesize that this intermediate state might be corresponding to the frequently flickering states that was observed in electrophysiology. So switching gears, I want to tell you, so these were the endogenous regulators of the channels. What about some exogenous modulators, especially lipidic modulators? So this work was done by Arvind Kumar, who was a postdoc in the lab, along with Kayla Aphrodite and Phil Biggins' group. So when you look at gating conformational changes, one thing that really pops out is that these cavities that I showed in the first slide, which are sites for binding of various modulators, so these cavities change volumes between closed and desensitized conformation. That might explain for some of the state-dependent binding of these ligands. So here I want to show you, so again, these are the cavities where a number of these modulators bind. So I want to highlight two of, actually one here in this case, is the glycine receptors are modulated by endocannabinoids. These are polyunsaturated fatty acid derived cannabinoids. For example, anandamide or arachidonyl ethanolamine potentiates glycine-1 currents. And also this sort of an effect is mimicked by THC or delta-9 tetrahydrocannabinol, which is the psychoactive ingredient in the cannabis plant. So you can see again potentiation. It has previously been proposed that THC binds within these transmembrane helices to bring about this potentiation. So again, so there is another modulators that I'll talk about is ivermectin, which binds into a site that is between the two subunits. So THC has this very nice and strong potentiation that you can see, and this potentiation is fully reversible, and you can recapture the activity. Like other allosteric modulators, at saturating concentration, you have this potentiation effect go away. So we solved a number of structures in the presence of varying concentrations of glycine. So unlike in the alpha 3, where at subsaturating concentrations we were at 100 micromolar, we captured multiple conformation. The sensitivity to glycine of alpha 1 is higher. So as you can see that at 100 micromolar, we simply have one desensitized conformation. So it was a challenging task for us to capture an open-like state in this case, or partially activated state. So in this case, so when we use THC, so here we are repeating this entire titration, but in the presence of 32 micromolar THC, we start to populate state that actually shift, that are somewhere in between the resting and the desensitized conformation. And in this pursuit, we actually captured an the transient open conformation. So this is a state that you have seen before. It's desensitized. Here is the desensitization gate closed in. And in molecular dynamic simulations, which allow you to look at permeation events across the membrane. So you can see that here this white patch suggests that there are fewer number of ions passing through this desensitized gate of the channel. So this is in the presence of THC, this channel's opening widens up, and then you start to see a more permeating channel. So again, the shift in equilibrium towards a more conducting conformation. So where is THC binding here? So THC, we have an additional density that we see likely corresponds to the THC molecule, which is in the same site that was previously proposed. It was proposed that THC interacts with the serine residue in the TM3 or the third transmembrane helix. Mutations to this have been shown before, and also we can see it in our hands that when you get rid of this serine side chain, then you lose or lower THC potentiation. So when we compare with and without THC, we notice that you have this binding pocket, but the structural changes are also seen along other TM helices such that it was sort of going towards the desensitized conformation. Particularly noteworthy are these rotameric reorientations of these side chains, which eventually physically couple to the desensitized gate. So we make mutations to each of these positions, and we notice that THC potentiation goes away, and in some cases pretty dramatic that there is absolutely no potentiation to THC. So a mechanism that we are proposing is THC binding right here, eventually reorients these aromatic residues to communicate to the desensitization gate, which stabilizes the open conformation. Phil Biggins' group, they took the THC coordinates, they docked it on the glycine alone state, and they looked at the stability, and you can see 3 out of 4 or 5 subunits holds on to THC, and in fact the conformational changes that the protein undergoes were similar to what we had observed in the cryo-EM structures. To the point that THC docked glycine structures move from these initial coordinates to start opening it up at the desensitized conformation. So to the last part, I want to tell you about another modulator, but in the context of a heteromeric glycine receptor. So this was a challenge. This work was done by Eric Gibbs and Emily Kruger, who is an MD Ph.D. student in the lab. So as we were working on this for several years, and we naively thought that we could use Gefrin, which binds to the intracellular domain. We could capture a heteromeric complex in the presence of its scaffolding protein, but also Gefrin would act as a fiducial marker, allowing us the alignment or identification of beta subunit in these. So we took a bit of a shortcut. We used just domain E, which is a dimerization domain, and this is a domain that has previously been shown to bind to the beta peptide region right here in the intracellular domain. And so we worked out a system to express all the 3 protein, and you can see it, and we got down to a pretty decent purification strategy. We could also see in gel filtration that we have a nice shift that indicates higher molecular weight, and we can also estimate the molecular weight by mass photometry. And then we started to image these. In the beginning, we started to see some additional densities that we had not seen in the intracellular domain before. But as we started to process it, we realized that the intracellular domain is so flexible, the 60 amino acids, that it swings slightly with the big, bulky Gefrin, and we really couldn't capture much. But we, in the process, were able to get some glimpses of the intracellular domain that I'll show you. But what was pretty neat is that the alpha and the beta subunits have different glycosylation sites. So these have these extensions that you see, and the beta subunit also has an N-terminal extension. So and the M4 lengths of alpha and beta subunit are different, and all these little differences were sufficient to unequivocally assign the subunits, the alpha and beta subunits, and determine stoichiometry. So here is a movie that it goes through multiple contouring so that you can see. So this is the alpha subunits are in light purple, and the violet is the beta subunit. And so this particular conformation was captured in the presence of strychnine, which is a highly toxic inhibitor of glycine receptors derived from strychnose plant. Here is the closed channel, and you can see the side chains that form the activation and the desensitization gate right there. This is the proline. So as we were looking through these structures, we noticed that the density for the intracellular domain, interestingly, is not placed at the center of the pore axis, but shifted away from the beta subunit. And we solved conformations of multiple, with multiple modulators in this, and we realized that the intracellular domain has a role to play. Besides all the other roles, at the gating part of it, it influences the overall movement of the transmembrane helices. So this is just the initial view. You can see the beta subunit, and this is the part of the intracellular domain. So we are really working very hard to tease this out and actually capture more of this domain. But here is the positional difference. So this is the bulk of the intracellular domain. And if you look at the movements within the transmembrane helices, the extent of movements of helices that are away from this bulky intracellular domain are much more than the movements that you see in the helices that are right beneath it. And the consequence of that is an asymmetric pore movement. And also we think that the anionic receptors have a different mode of gating within the intracellular domain. This is because previous work from our lab and others have shown this is from serotonin receptors as a channel gates. You have the opening of these intracellular domain leading to creation of these portals through which ions go through. In this case, it's not a direct portal, and this is a pretty interesting architecture that we want to dig further into. And so this is the asymmetry of the pore that I'm talking about. So having an ICD or intracellular domain right here, this is no more a symmetrical movement of the pore helices during gating. There were differences in the lipid densities. There were differences in the protein architecture between alpha and beta subunits. Previously, it has been shown that certain modulators do not bind to the beta subunit, and ivermectin was proposed to be one of them. So ivermectin it's a pretty strong potentiator of glycine receptors, potentiates at low glycine concentrations, unique in that at higher concentration it can act as an activator. So it's known that it binds within the transmembrane domain. Previously, Eric Goh's lab has shown this. So this is where ivermectin binds. It binds at the interface between the two subunits and directly contacts M2, which forms the pore of the channel. And then it sort of wedges in and activates the channel. So it was previously suggested that, so as it inserts, this is looking at it from the top, there is an alanine residue in the alpha subunit, whereas this residue is isoleucine in the beta subunit. So because of this bulkiness, it was proposed that it likely doesn't bind this beta subunit. And again, I think a nice feature about this is that ivermectin is an antiparasitic drug, and it in nematodes where there is a channel that is glutamate gated, chloride channel, so it is activated very strongly by ivermectin, and this residue right here is a glycine. So you make this residue smaller, you have a greater potentiation with ivermectin. So again, but what we found is that ivermectin binds into all of the interfaces, but has a unique orientation in the beta subunit interface, comes because of this isoleucine pushing over it and wedging out the ivermectin molecule. So here is a top view that you can see. This is in the alpha versus beta subunit. Molecular dynamic simulations show that this binding mode has a weaker affinity for glycine. And so we then asked, okay, if this is the case, if ivermectin is a ligand that has its own activation pathway, so instead of activation originating at the level of the extracellular domain, moving into the transmembrane region, now you have a path that goes from transmembrane domain. What is it? So can we use this to understand whether there are binding affinity differences? Can we capture intermediates? Because we've been able to only get these states, either closed or open or desensitized. So what we decided to do was we added strychnine, which is going to clamp down the path from here to here, and then we add ivermectin, which challenges the path from the transmembrane to the extracellular domain to basically look at binding modes and also look at intermediates if possible. So in this pursuit, so we have ivermectin, with strychnine at saturating concentrations, and then we titrate ivermectin out. And in doing so, what we start to see is this is the benchmarking. This is in the presence of glycine ivermectin. You see these binding modes, alpha-alpha interfaces, alpha-beta and beta-alpha interface. You can see this distinct mode here. So as you start to decrease ivermectin concentration, we start to lose on the occupancy first in the beta interface, beta-alpha interface, then in alpha-beta, and then eventually in the alpha-alpha interfaces. Suggesting a high-affinity binding mode here, which sort of shifts down as we go towards beta interfaces. And then we can also look at the conformation. So this is simply looking at projection. This is resting. This is activated state. And thinner these helices and these strands, it means that they look more like strychnine. As they become more red and thicker, they look more like the open conformation. So at this low concentration of ivermectin, you have a resting or a strychnine-bound state. But as you increase concentrations, you start to move towards the activated state right here. But what's interesting is most of the conformations within the transmembrane domain has already happened. So even with partial ivermectin occupancies, you have a fully activated state. But the response in the extracellular domain is more nuanced. As you can see, some of these regions are activated, some of them are not, likely suggesting intermediates within the gating pathway. So here is a movie that summarizes this. So you have this movement. There is no ivermectin occupancy yet. You can see there is not much change. Then as you move, you can see the first ivermectin comes in, and then as you start to see higher binding affinity modes, then the channel moves towards. But if you align them, you either see they're activated or they're not. So there is a full cooperativity in this case. On the other hand, things are more graded here in the extracellular domain. So this is strychnine, and you can also see as you increase ivermectin, strychnine's orientation starts to change and move towards low affinity modes. So again, so these are some of the different, we believe, gating intermediates in this activation pathway. Another way to represent here, this is just along the length of the sequence. So changes here in the transmembrane domain are almost all or none, whereas here you see very many intermediates within the extracellular domain. So this is sort of, concludes what I wanted to say here. We are just starting to understand the basis for the subtype specificity. We are still scratching the tip of the iceberg here. And hopefully we can use these understanding to look at other modulators. So a general summary is that these cryo-EM structures from homeric and heteromeric channels reveal these intricate movements that underlie functional behaviors and modulations, and we can start to tease out asynchronous movements that are part of these individual domains. And I think the big part of, we want to understand modulation, we want to understand how they're modulated by lipids and lipidic modulators and drug and compounds that have good therapeutic profiles. So with that, I want to acknowledge the people who have done all of this work, particularly all the past members who have moved on, and this is our current team, and I've acknowledged individual people as we went through. This is the work of the 5-3 team that I didn't talk about. Our collaborators, cryo-EM access. We're fortunate to have local access, but we routinely use national centers for our work, support from NIGMS, and thank you all for listening. Happy to take questions.
I encourage people to use the microphones. There's no questions online yet. We do have a robust audience. I will say the CME code again, 57663. And please use the mics if any.
Sudha, that was a beautiful talk. I have a question. I was very intrigued by sort of this change in the cavity volumes that you alluded to. And I'm imagining that as these proteins in general undergo conformational changes, not only do you change the volumes of these cavities, you also potentially change the surface chemistry of these cavities. So in principle, that means that opens up the possibility of these proteins being regulated by phospholipids that are in the membrane. Is there any way to tease this apart? Do you think this is just a fantasy? Can you sort of educate
Anubhav.
me on
Yes.
this.
Yeah, no, I mean, absolutely. Fascinating question. So, and also a challenge for a structural biologist. I mean, this is our field, and we struggle to tease this apart, right? So these, I mean, what you point out is very important. It's not just the volume, it's the chemistry of this. So some of these pockets are hydrophobic. As they move out and they reorient the helices, the hydrophilicity of those pockets change. And once the channel under normal gating conditions, as they open and these cavities open up, it's not in vacuum. It's, you have lipids getting there, and those lipids are natural regulators of channel function. And now you put a modulator that looks like some of these lipids. How do you distinguish between that? So I think that's a challenge. And also when you work with a minimalistic system, we really don't have those lipids when we put the modulator in. So their affinities are probably different. So I think those are some of the challenges. I think, can we use native mass spec to start identifying what those lipids are, and then can we start incorporating them into our structural studies? But I think interesting and challenging pursuit there.
A couple questions are coming in online. Okay, so do you think the pH dependence of zinc activation on the glycine receptor is due to the ionization state of histidine, which coordinates to the high affinity zinc state and to the low affinity zinc state?
Yeah, so there are great questions. So about the pH effects and the ionizable residues, and what are the residues that are mediating this? It's unclear at this point. But there are several residues that have been implicated in pH dependence. So histidines in the potentiating sites are likely to be some of them, because then again, when you have acidic pH, zinc modulation goes away, zinc potentiation goes away. So it is likely that those histidines play a role. Also, interestingly, some threonines have been implicated, that you make mutations to those threonines, two of them, that they eliminate pH or acidic pH inhibition. It's unclear what threonines are doing there, but it's also possible that it's an allosteric effect, that you make a mutation to the threonine, it is changing the way the histidine is seeing another interaction, and it could be possible because of that. But for sure, the potentiating residues have a role directly.
Okay, another question. Is the binding constant Kd known for the zinc or other ligands?
So the binding, so for all the ligands that I was talking about, again, the Kds vary quite a bit. So for the zinc, at the lower, so for sure, there are low micromolars for the potentiating sites and higher for the inhibitory site, getting closer. I mean, again, it depends on the type of subtype that you're looking at. The alpha 3 is in the 300 micromolar range for the inhibitory one.
Okay. Dr. Lush, any profound closing comments?
So do heavy metals bind to the zinc sites?
Also, yes.
Does that have anything to do with lead or mercury neurotoxicity?
Yeah, I mean, it is possible. Again, the site is prime for binding other divalents, and it's known that some of these divalents have. But in terms of whether they lead to neurotoxicity and that kind of thing, unclear, but it is possible. I mean, that site, it's not exclusive to zincs, as you can see from the coordination. Right. So, yeah. But its direct role, not sure. Yeah.
Fantastic. If there are no other questions, then let's thank Sudha for a fantastic talk. Oh, and don't forget there's a reception outside, so please join us.